In most energy transformations, ordered forms of energy are at least partly converted to heat. About 25% of the chemical energy stored in the fuel tank of an automobile is transformed into the motion of the car. The remaining 75% is lost from the car engine as heat, which dissipates rapidly through the surroundings. In performing various kinds of work, living cells unavoidably convert organized forms of energy to heat. This makes a room crowded with people uncomfortably warm. In machines and organisms, even energy that performs useful work is eventually converted to heat. The organized energy of an automobile's forward movement becomes heat when the friction of the brakes and tires stops the car. Conversion to heat is the fate of all the chemical energy. Conversion of other forms of energy to heat does not violate the first law of thermodynamics. Energy has been conserved, because heat is a form of energy, albeit in its random state. By combining the first and second laws of thermodynamics, we conclude that the quantity of energy in the universe is constant, but its quality is not. In a sense, heat is the lowest rate of energy, because it is an uncoordinated movement of molecules that many systems cannot harness in order to perform work. A system can only put heat to work when there is a temperature difference that results in the heat flowing from a warmer location to a cooler one. If temperature is uniform throughout a system, as it is in a living cell, then the only use for heat energy is to warm a body of matter, such as an organism. Thus, an organism takes in organized forms of matter and energy from the surroundings and replaces them with less ordered forms. For example, animal obtains starch, proteins, and other complex molecules from the food it eats, and in turn releases carbon dioxide and water, relatively small and simple molecules that store less chemical energy than the food. The depletion of chemical energy is accounted by heat generated during metabolism. Cells create ordered structures from less organized starting materials. For example, amino acids are ordered into the specific sequences of polypeptide chains. Complex organisms evolve from simpler ancestors. But, the high degree of organization in no way violates the second law, because the entropy of a particular system, such as an organism, may actually decrease, as long as the total entropy of the universe, the system plus its surroundings, increases. Thus, organisms are islands of low entropy in an increasingly random universe. The evolution of biological order is perfectly consistent with the laws of thermodynamics. EU. Thermodynamic principles in biology. Free energy change. If we consider a system consisting of two compounds A and B which react to give C A plus BC by reacting, the system can exchange work or energy, heat, light with the immediate surroundings. For example, it can receive energy and produce an equivalent work. At the time of mixing, A plus B represents the initial state, and C, the product formed, represents the final state. Each of these two states is characterized by an internal energy level U1 and 2, respectively, which depends on the chemical nature of the constituents, with their atoms, their bonds, their concentrations and their position in space, absolute temperature T, pressure P, volume 5. The internal range, denoted by increment U, resulting from a change of molecular structures, new bonds and different distribution of electrons depends only on the initial state and final state. Increment U equals U2 U1. The maximum quantity of energy which can be made available to accomplish a work by the transformation of A plus B into C in a constant pressure system, as in the case of many biological reactions, there can be volume changes in either a liberation or an absorption of heat. The work done by pressure is equal to P V2 V1, i.e., P increment V. The heat of reaction first defined by increment Q is equal to the internal energy change plus the work done. Increment Q equals increment U plus P increment V. Increment H equals increment U plus P increment V1. Given temperature, because we know that PV equals nRT, N equals number of moles, R equals gas constant, T equals temperature, in degrees Kelvin. Increment H is the enthalpy change of the system. Increment H, like increment Q, is mostly expressed in kcal per mole, but ought to be expressed in joule per mole. 1 calorie equals 4.18 joules. Increment H can be measured in a calorimeter. If increment H less than 0, the reaction is exothermic, heat is liberated, and if increment H greater than 0, the reaction is endothermic, the system absorbs heat. In the past, it was thought that the knowledge of the sign of increment H is sufficient to predict whether a reaction is spontaneous or not, and it was said that an exothermic reaction is spontaneous, while an endothermic reaction is not. It found that, while these predictions are correct in most cases, there are exceptions, and that some spontaneous reactions are, in fact, accompanied by absorption of heat by the molecules. One is therefore led to consider the entropy of the system denoted as a function which measures the disorder of a system and the variations of which, increment S equals S2 S1, depends, like increment U, on the initial state and the final state. In order to pass from a first stable state, the initial state, to a new stable state, the final state, and one must upset the structures of molecules A and B for the reaction to take place. The greater increase in molecular disorder, the greater the entropy. For example, entropy increases while passing from a given protein to the constituent amino acids in free state. Entropy is related to the complexity of the molecules. The larger the number of atoms, the larger the number of complicated chemical groups, the larger the number of possible structures and the greater the entropy. In a reversible transformation at constant pressure, increment Q equals T increment S. One can then write increment equals G plus increment Q equals increment G plus T increment S2, or increment G, free enthalpy change, equals increment HT increment S3. In a reversible transformation at constant volume, increment V equals O, the free energy change is F equals increment UT increment S4. In biochemistry, it may be admitted that in most cases increment V equals 0, because the volume changes for reactions taking place in solution are negligible. In increment G, it is, therefore, admitted that P increment V equals 0, so that increment G becomes, like increment F, equation 4 equal to increment UT increment S, and, therefore, increment F becomes increment G. This is particularly true if the reactions involve liquids and solids in solution, and no gas, at physiological temperatures which are low. 3 cases are possible. When increment G less than 0, the reaction is exergonic, it can be spontaneous, and free energy is available. On the contrary, if increment G greater than 0, the reaction is endergonic, free energy must be supplied to the system for the reaction to take place. The reaction will take place only coupled with another exergonic reaction whose negative increment G is greater in absolute value than the positive increment G of the endergonic reaction. Many biochemical reactions are reversible. Increment G equals 0 when dynamic equilibrium is reached. This is very interesting, because, for very small variations of active masses of the reaction's participants, the reaction can take place in either direction, without energy problem, as increment G will vary in either direction. It may note increment S, as T is comparatively low in biochemistry, the term T increment S can be neglected and then increment G equals increment H, see equation 2, in other word, in this case, the terms exothermic and exergonic, on the one hand, and endothermic and endergonic, on the other, are similar. Application to chemical reactions. We consider in solution at 25 degrees Celsius under a pressure of 1 atmosphere, each in a concentration of 1 mole liter of solvent 1 mole and which can react completely, giving C plus D, which will, therefore, be present in the same concentration after total transformation. The free energy change is called standard and denoted increment GO. If GO less than 0, the reaction is exergonic. It can take place spontaneously, but this is not necessarily the case even if the free energy change is very large. In fact, increment GO expresses the free energy change between an initial state and a final state. However, increment GO gives no information on reaction velocity. A catalyst, an enzyme for example 1E might be needed to trigger the reaction and adjust its velocity without altering its thermodynamic characteristics. Despite the high negative of increment GO, 686 kilocalories per mole, of the total oxidation reaction of G1 glucose, C6H12O6 plus 6O2 plus 6CO2 plus 6H2O, this substance can be left for years in the presence of the oxygen of air, without any oxidation taking place. On the one hand, when GO indicates a standard state, it does not take into consideration the special circumstances which may prevail, particularly in the biological medium and condition and activities of compounds, pH, etc. Normally reaction proceeds up to the equilibrium state and increment G takes this equilibrium state into constant, but the reaction can proceed up to completion if the equilibrium state is continuously broken because the reaction products are progressively consumed by a following reaction. This is what happens in metabolic chains here. In fact, a general state of equilibrium is observed. On the other hand, reactions which at the limit can be reversible in vitro can be reversed in vivo because of metabolic conditions. Actually, the free enthalpy change increment G or free energy change increment F will always depend on metabolic circumstances and will be related to equilibrium states in vivo. GO depends on the equilibrium constant K of the reaction. Increment GO equals RT lane K. And that in given circumstances, the effective increment G of a reaction is opening square bracket closing square bracket 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 0 lane AB CD increment G equals increment G plus RT. First factor, second factor. For a reversible reaction A plus B if and only if C plus D proceeding from left to right, the increment G of the reverse reaction will have the opposite sign. Therefore, increment G, on the one hand, the increment GO which characterizes a given reaction, and on the other hand the second factor, characteristic of given circumstances and hence related to concentration, activity, dissociation or protonation or the reaction participants, etc. When the rate of activities of the second factor becomes equal to 1, the second factor becomes 0 and increment G equals increment GO. It must be noted G and increment GO correspond to pH equals 0 and ambient temperature while increment G and increment G all represent the values in the physiological conditions pH near 7 physiological temperature, 30 to 37O to be specified in each case. Let us examine the case of a reaction whose increment GO equals minus 3 kilocalories per mole. R equals 1.987 calories per mole degree, that at 25 degrees Celsius, T equals 273 plus 25 equals 2980 K and that in K equals 2.30 log K. We can write increment GO equals minus 3000 calories per mole equals minus 1.987 x 298 by 2.30 log K, therefore log K equals minus 3000 equals 2.2. Minus 1362 i.e. approximately equal to 2, which corresponds to value K equal to about 100. The reaction has an increment GO equals plus 3 kilocalories per mole, then K will be approximately equal to 0.01. In other words, the reaction will practically not take place because equilibrium will be reached as soon as only 1% of the reactants will have been transformed. It follows the increment GO can be calculated if one knows K, i.e., if one can measure the concentrations of the reactants and reaction products, this is possible, except when K is extremely small or extremely large, because, then, the concentrations of the reaction products or reactants are too low to be determined accurately. Table gives the values of increment GO for series of values of K from 10 to 3 to 10.6. It is clear the reactions whose equilibrium constant is greater than 1 are accompanied by a decrease of free energy. This energy becomes available. These are exergonic reactions. On the contrary, if K is less than 1, the reaction is endergonic. Log increment G equals minus 1392 log K in calorie per mole. E3. Glycolysis. The larger the number of atoms, the larger the number of complicated chemical groups, the larger the number of possible structures and the greater the entropy. In a reversible transformation at constant pressure, increment Q. E3. Glycolysis. Glycolysis is derived from the Greek words glycos, sweet or sugar, lysis dissolution. It is a universal pathway in the living cells. This pathway is often referred to as Embden-Meyerhof pathway, EM pathway, in honor of the two biochemists who made a major contribution to the knowledge of glycolysis. Glycolysis is defined as the sequence of reactions converting glucose or glycogen to pyruvate or lactate with the production of ATP. Salient features. Glycolysis takes place in all cells of the body. The enzymes of this pathway are present in the cytosomal fraction of the cell. 2. Glycolysis occurs in the absence of oxygen, anaerobic, or in the presence of oxygen, aerobic. Lactate is the end product under anaerobic condition. In aerobic condition, pyruvate is formed, which is then oxidized to CO2 and H2O. 3. Generally, Embden-Meyerhof pathway is an emergency energy-yielding pathway for cells in the absence of oxygen. 4. The occurrence of glycolysis is a prerequisite for the aerobic oxidation of carbohydrates. The latter takes place in the cells possessing mitochondria. 5. Glycolysis is a major pathway for ATP synthesis in tissues lacking mitochondria, e.g. erythrocytes, cornea, lens, etc. 6. In some others which have relatively few mitochondria, e.g. testes, leukocytes and kidney medulla, glycolysis is significant for ATP production. 7. Lactate is the end product under anaerobic. This pathway is often referred to as Embden-Meyerhof pathway, EM pathway, in honor of the two biochemists who made a major contribution to the knowledge of glycolysis. Glycolysis is defined as the sequence of reactions converting glucose or glycogen to pyruvate or lactate with the production of ATP. Salient features. 1. Glycolysis takes place in all cells of the body. The enzymes of this pathway are present in the cytosomal fraction of the cell. 2. Glycolysis occurs in the absence of oxygen, anaerobic, or in the presence of oxygen, aerobic. Lactate is the end product under anaerobic condition. In the aerobic condition, pyruvate is formed, which is then oxidized to CO2 and H2O. 3. Generally, Embden-Meyerhof pathway is an emergency energy-yielding pathway for cells in the absence of oxygen. 4. The occurrence of glycolysis is a prerequisite for the aerobic oxidation of carbohydrates. The latter takes place in the cells possessing mitochondria. 5. Glycolysis is a major pathway for ATP synthesis in tissues lacking mitochondria, e.g. erythrocytes, cornea, lens, etc. 6. In other tissues which have relatively few mitochondria, e.g. testes, leukocytes and kidney medulla, glycolysis is significant for ATP production. 7. Glycolysis is very essential for brain, which is dependent on glucose for energy. Glucose in brain has to undergo glycolysis before it is oxidized to CO2 and H2O. 8. Glycolysis anaerobic may be summarized by the net reaction. Glucose plus 2 ADP plus 2 pi. 2 lactate plus 2 ATP. 9. The intermediates formed in glycolysis are useful for the synthesis of non-essential amino acids and glycerol, the latter used for fat formation. 10. Reversal of glycolysis along with the alternate arrangements made at the irreversible steps will result in the synthesis of glucose gluconeogenesis. Reactions of glycolysis. The sequence of reactions of glycolysis are given in Figure 1. The pathway can be divided into 3 distinct phases. A. Energy investment phase or priming stage. B. Splitting phase. C. Energy generation phase. A. Energy investment phase. 1. Glucose is phosphorylated to glucose 6-phosphate by hexokinase or glucokinase. This is an irreversible reaction, dependent on ATP and Mg2+. The enzyme hexokinase is present in almost all the tissues. It catalyzes phosphorylation of various hexoses, fructose, mannose, etc., has low Km for substrates, about 0.1 m, and is inhibited by glucose 6-phosphate. Cokinase present in liver, catalyzes the phosphorylation of only glucose, has high Km for glucose 10 m, and is not inhibited by glucose 6-phosphate. Due to high affinity, low Km, glucose is utilized by hexokinase even at low concentration, whereas glucokinase acts only at higher levels of glucose, i.e., after a meal when blood glucose concentration is above 100 mg per deciliter. Glucose 6-phosphate is impermeable to the cell membrane. It is a central molecule with a variety of metabolic fates: glycolysis, glycogenesis, gluconeogenesis, and pentose phosphate pathway. 2. Glucose 6-phosphate undergoes isomerization to give fructose 6-phosphate in the presence of the enzyme phosphohexose isomerase and Mg2+. 3. Fructose 6-phosphate is phosphorylated to fructose 1,6-bisphosphate by phosphofructokinase PFK. This is an irreversible and regulatory step in glycolysis. PFK is an allosteric enzyme, the activity of which is controlled by several allosteric modulators. B. Splitting phase. 4. The 6-carb-fructose 1,6-bisphosphate is split, hence the name glycolysis, to two 3-carbon compounds, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate by the enzyme aldolase, fructose 1,6-bisphosphate aldolase. 5. The enzyme aldolase isomerase catalyzes the reversible interconversion of glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Thus, 2 molecules of glyceraldehyde 3-phosphate are obtildaine from 1 molecule of glucose. This isomerase enzyme is inhibited by bromohydroxylacetone phosphate. C. Energy generation phase. 6. Glyceraldehyde 3-phosphate dehydrogenase converts glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. This enzyme brings about 2 reactions, namely oxidation, where the hydrogens are transferred to NAD+, and phosphorylation of substrate adding 1-phosphate moiety. This step is important as it is involved in the formation of NADH plus H plus and a high-energy compound 1,3-bisphosphoglycerate. Lodoacetate and arsenate inhibit the enzyme glyceraldehyde 3-phosphate dehydrogenase. In aerobic condition, NADH passes through the electron transport chain and 6-ATP, 2x3 ATP, are synthesized by oxidative phosphorylation. 7. The enzyme phosphoglycerate kinase acts on 1,3-bisphosphoglycerate, resulting in the synthesis of ATP and formation of 3-phosphoglycerate. is a good example of substrate-level phosphorylation, since ATP is synthesized from a substrate without the involvement of electron transport chain. Phosphoglycerate kinase reaction is reversible, a rare example among the kinase reactions. 8. 3-phosphoglycerate is converted to 2-phosphoglycerate by phosphoglycerate mutase. This is an isomerization reaction, involving the transfer of phosphate group from position 3 to position 2 in the substrate. The enzyme requires Mg2+. 9. The high-energy compound nisphenol pyruvate is generated from 2-phosphoglycerate by the enzyme enolase. This enzyme requires Mg2+ or Mn2+ and is inhibited by fluoride. Glucose estimation in the laboratory. Fluoride is added to the blood to prevent glycolysis by the cells, so that blood glucose is correctly estimated. 10. The pyruvate kinase catalyzes the transfer of high-energy phosphate from phosphoenol pyruvate to ADP, leading to the formation of ATP. This step also is a substrate-level phosphorylation. Pyruvate kinase requires K+ in either Mg2+ or Mn2+. This reaction is irreversible. Conversion of pyruvate to lactate significance. The fate of pyruvate produced in glycolysis depends on the presence or absence of oxygen in the cells. In anaerobic conditions, lack of O2, pyruvate is reduced by NADH to lactate in presence of the enzyme lactate dehydrogenase, competitive inhibitor oximate. NADH utilized in this step is obtained from the reaction catalyzed by glyceraldehyde 3-phosphate dehydrogenase. The lactate allows the regeneration of NAD+, which can be reused by glyceraldehyde 3-phosphate dehydrogenase so that glycolysis proceeds even in the absence of oxygen to supply ATP. Uninterrupted glycolysis is very essential in skeletal muscle during laborious exercise where oxygen supply is very limited. Glycolysis in the erythrocytes leads to lactate production, since mitochondria, the centers for aerobic oxidation, are absent. Brain, retina, skin, renal medulla and gastrointestinal tract derive most of their energy from glycolysis. Production of ATP in glycolysis. In aerobic conditions, 2 ATP are synthesized while, under aerobic conditions, 8 or 6 ATP are synthesized depending on the shuttle pathway that operates. Glycolysis and shuttle pathways. Glycolysis takes place in the cytosol. NADH produced by the enzyme, glyceraldehyde 3-phosphate dehydrogenase, is utilized later by the enzyme lactate dehydrogenase and NAD+ is regenerated. So far as the synthesis and utilization of reducing equivalents are coupled, glycolysis proceeds uninterrupted and the end product is lactate. The perfect coupling is possible only in cells that lack mitochondria or under anaerobic conditions. However, in the presence of mitochondria and oxygen, the NADH produced in glycolysis can participate in the shuttle pathways for the synthesis of ATP. If the cytosolic NADH uses malate aspartate shuttle, 3 ATP are generated from each molecule of NADH. This is in contrast to glycerol phosphate shuttle that produces only 2 ATP. Irreversible steps in glycolysis. Most of the reactions of glycolysis are reversible. However, the 3 steps catalyzed by the enzymes hexokinase or glucokinase, phosphofructokinase and pyruvate kinase are irreversible. These 3 stages mainly regulate glycolysis. The reversal of glycolysis, with alternate arrangements made at the 3 irreversible stages, leads to the synthesis of glucose from pyruvate gluconeogenesis. Regulation of glycolysis. Enzymes namely hexokinase and glucokinase, phosphofructokinase and pyruvate kinase, catalyzing the irreversible reactions, regulate glycolysis. Hexokinase is inhibited by glucose 6-phosphate. This enzyme prevents the accumulation of glucose 6-phosphate due to product inhibition. Glucokinase, which specifically phosphorylates glucose, probably through the involvement insulin, induces glucokinase. Phosphofructokinase PFK is the most important regulatory enzyme in glycolysis. This enzyme catalyzes the rate-limiting committed step. PFK is an allosteric enzyme regulated by allosteric effectors. AP, citrate and H+ ions, low pH, are the most important allosteric inhibitors, whereas fructose 2, 6-bisphosphate, AMP and pi are the allosteric activators. Role of fructose 2, 6-bisphosphate. Fructose 6-bisphosphate F2, 6BP is considered to be the most important regulatory factor, activator, for controlling PFK and, ultimately, glycolysis in the liver. F2, 6BP is synthesized from fructose 6-phosphate by the enzyme phosphofructokinase called PFK2. PFKL is a glycolytic enzyme. F2, 6BP is hydrolyzed by fructose 2, 6-bisphosphatase. Synthesis and degradation of F2, 6BP is brought out by a single enzyme, same polypeptide with 2 active sites, which is referred to as bifunctional enzyme. IAC, combined name of PHOS-PHOFRUCTOKINASE2, fructose 2, 6-bisphosphatase is used to refer to the enzyme that synthesizes and degrades F2, 6BP. Activity of PFK2 and fructose 2, 6-bisphosphatase is controlled by covalent modification which, in turn, is regulated by cyclic AMP. CAMP is a second messenger for certain of hormones. Cyclic AMP is about dephosphorylation of the bifunctional enzyme, resulting in inactivation of active site responsible for the synthesis of F2, 6BP but activation of the active site responsible for the hydrolysis of F2, 6BP. Pyruvate kinase also regulates glycolysis. This enzyme is inhibited by ATP and activated by FLAT, 6BP. Pyruvate kinase is active in dephosphorylated state and inactive B in phosphorylated state. Inactivation of pyruvate kinase by phosphorylation is brought about by CAMP-dependent protein kinase. The hormone glucagon inhibits hepatic glycolysis by this mechanism. Pasteur effect. The inhibition of glycolysis by oxygen, aerobic condition, is known as Pasteur effect. This effect was discovered by Louis Pasteur, more than a century ago, while studying fermentation by yeast. He observed that when anaerobic yeast cultures metabolizing yeast were exposed to air, the utilization of glucose decreased by nearly 7-fold. In the aerobic condition, the levels of glycolytic intermediates from fructose 1, 6-bisphosphate onwards decrease while the earlier intermediates accumulate. This clearly indicates that Pasteur effect is due to the inhibition of the enzyme phosphofructokinase. The inhibitory effect of citrate and ATP produced in the presence of oxygen on phosphofructokinase explains the Pasteur effect. Crabtree effect. The phenomenon of inhibition of oxygen consumption by the addition of glucose to tissues having high aerobic glycolysis is known as Crabtree effect. Basically, this is opposite to that of Pasteur effect. Crabtree effect is due to increased competition of glycolysis for inorganic phosphate Pi and NAD+, which limits their availability for phosphorylation and oxidation. Rappaport-Lubering cycle. This is a supplementary pathway to glycolysis which is operative in the erythrocytes of man and other mammals. Rappaport-Lubering cycle is mainly concerned with the synthesis of 2,3-bisphosphoglycerate 2,3-BPG in the RBC1, 3-bisphosphoglycerate 1,3-BPG produced in glycolysis is converted to 2,3-BPG by the enzyme 2,3-bisphosphoglycerate mutase figure 13.5. 2,3-BPG is hydrolyzed to 3-phosphoglycerate by bisphosphoglycerate phosphatase. Note, there is a difference between the usages bisphosphate and diphosphate. A bisphosphate has two phosphates held separately, e.g. 2,3-BPG in contrast to diphosphate, e.g. ADP, where the phosphates are linked together. Believe that bisphosphoglycerate mutase is a bifunctional enzyme with mutase and phosphatase activities catalyzed by two different sites present on the same enzyme. About 15-25% of the glucose converted to lactate in erythrocytes goes via 2,3-BPG synthesis. Significance of 2,3-BPG. 1. Production of 2,3-BPG allows the glycolysis to proceed without the synthesis of ATP. This is advantageous to erythrocytes since glycolysis occurs when the need for ATP is minimal. Rappaport-Lubering cycle is, therefore, regarded as a shunt pathway of glycolysis to dissipate or waste the energy not needed by erythrocytes. 2,3-BPG, however, is not a waste molecule in RBC. It combines with hemoglobin, Hb, and reduces Hb affinity with oxygen. Therefore, in the presence of 2,3-BPG, oxyhemoglobin unloads more oxygen to the tissues. Increase in erythrocytes 2,3-BPG is observed in hypoxic condition, high altitude, fetal tissues, anemic conditions, etc. In all these cases, 2,3-BPG will enhance the supply of oxygen to the tissues. 3. Glycosis in the erythrocytes is linked with 2,3-BPG production and oxygen transport. Of the enzyme hexokinase, glucose is not phosphorylated, hence the synthesis and concentration of 2,3-BPG are low in RBC. The hemoglobin exhibits high oxygen affinity in hexokinase effective patients. On the other hand, in the patients with pyruvate kinase deficiency, the level of 2,3-BPG in erythrocytes is high, resulting in low oxygen affinity. Conversion of pyruvate to acetyl-CoA. Pyruvate is converted to acetyl-CoA by oxidative decarboxylation. This is an irreversible reaction, catalyzed by a multi-enzyme complex, known as pyruvate dehydrogenase complex, PDH, which is found only in the mitochondria. High concentrations of PDH are found in cardiac muscle and kidney. Enzyme PDH requires 5 cofactors coenzymes, namely TPP, lipomide, FAD, coenzyme A and NAD+. Epomide contains lipoic acid linked to pound amino group of lysine. The overall reaction of PDH is PDH. Pyruvate plus NAD++ CoA. Acetyl-CoA plus CO2 plus NADH plus H plus reactions of PDH complex. The C reactions brought about by different enzymes of PDH complex in association with the coenzymes is depicted in figure. Pyruvate is decarboxylated to give hydroxyethyl TPP, catalyzed by PDH decarboxylase activity. Dihydroapoyl transacetylase brings about the formation of acetyl-lipomide from hydroxyethyl TPP and then catalyzes the transfer of acetyl group to coenzyme to produce acetyl-CoA. The cycle is complete when reduced lipomide is converted to oxidized lipomide by dihydroapoyl dehydrogenase, transferring the reducing equivalents to FAD. FADH2, in turn, transfers the reducee NAD+ to give NADH plus H+, which can pass through the respiratory chain to give 3 ATP, 6 ATP from 2 moles of pyruvate formed from glucose by oxidative phosphorylation. The intermediates of PDH catalyzed reaction are not free but bound with enzyme complex. In mammals, the PDH complex has an approximate molecular weight of 9 x 10^6. It contains 60 molecules of dihydroapoyl transacetylase and about 20 to 30 molecules each of the other two enzymes, pyruvate dehydrogenase and dihydroapoyl dehydrogenase. A comparable enzyme with PDH is acetoglutarate dehydrogenase complex of citric acid cycle, which catalyzes the oxidative decarboxylation of alpha-ketoglutarate to succinyl-CoA. Biochemical importance of PDH. 1. Lack of TPP due to deficiency of thiamine inhibits PDH activity, resulting in the accumulation of pyruvate. 2. In the thiamine-deficient alcoholics, pyruvate is rapidly converted to lactate, resulting in lactic acidosis. 3. In patients with inherited deficiency of PDH, lactic acidosis, usually after glucose load, is observed. 4. PDH activity can be inhibited by arsenic and mercuric ions. This is brought about by binding of these ions with SH groups of lipoic acid. Note, pyruvate is a key metabolite. Besides its conversion to acetyl-CoA, utilized in a wide range of metabolic reactions, citric acid cycle, fatty acid synthesis, etc., pyruvate is a good substrate for gluconeogenesis. E4. Biological oxidation. Oxidation is defined as the loss of electrons and reduction as the gain of electrons. This may be illustrated by the interconversion of ferrous ion Fe2+ to ferric ion Fe3+. The electron lost in the oxidation is accepted by an acceptor, which is said to be reduced. Thus the oxidation reduction is a tightly coupled process. The general principle of oxidation reduction is applicable to biological systems also. The oxidation of NADH to NAD+ coupled with the reduction of FMN to FMNH2 is illustrated. In the above illustration, there are two redox pairs NADH, NAD+ and FMN, FMNH2. The redox pairs differ in their tendency to lose or gain electrons. Redox potential EO, the oxidation reduction potential, or, simply, redox potential, is a quantitative measure of the tendency of a redox pair to lose or gain electrons. The redox pairs are assigned specific standard redox potential EO volts at pH 7.0 and 25 degrees Celsius. The redox potentials of some biologically important redox systems are given in Table Point 3. The more negative redox potential represents a greater tendency of reductant to lose electrons. On the other hand, a more positive redox potential indicates a greater tendency of oxidant to accept electrons. The electrons flow from a redox pair with more negative EO to another redox pair with more positive EO. The redox potential EO is directly related to the change in the free energy increment GO. Electron transport chain. The energy-rich carbohydrates, particularly glucose, fatty acids, and amino acids, undergo a series of metabolic reactions and, finally, get oxidized to CO2 and H2O. The reducing equivalents from various metabolic intermediates are transferred to coenzymes NAD+ and FAD to produce, respectively, NADH and FADH2. The latter reduced coenzymes pass through the electron transport chain, etc., or respiratory chain and, finally, reduce oxygen to water. The passage of electrons through the etc. is associated with the loss of free energy. A part of this free energy is utilized to generate ATP from ADP in Fig. Point 3. The powerhouses of cell. The mitochondria are the centers for metabolic oxidative reactions to generate reduced coenzymes NADH and FADH2, which, in turn, are utilized in etc. to liberate energy in the form of ATP. For this reason, mitochondrion is appropriately regarded as the powerhouse of the cell. Mitochondrial organization. The mitochondrion consists of 5 distinct parts. Rhizo, the outer membrane, the inner membrane, the intermembrane space, the cristae, and the matrix. The electron transport chain and ATP synthesizing system are located on the inner mitochondrial membrane, which is a specialized structure, rich in proteins. It is impermeable to ions H+, K+, Na+, and small molecules ADP, ATP. This membrane is highly folded to form cristae. The surface area of inner mitochondrial membrane is greatly increased due to cristae. The inner of the inner mitochondrial membrane possesses specialized particles that look like lollipops, the phosphorylating subunits, which are the centers for ATP production. Mitochondrial matrix. The interior ground substance forms a matrix of mitochondria. It is enzymes responsible for the citric acid cycle, beta oxidation of fatty acids and oxidation of amino acids. Structural organization of respiratory chain. The inner mitochondrial membrane can be disrupted into 5 distinct respiratory or enzyme complexes, denoted as complex I, II, III, IV and V, Fig. 6. The complexes I, IV are carriers of electrons while complex V is responsible for ATP synthesis. Besides these enzyme complexes, there are certain mobile electron carriers in the respiratory chain. These include NADH, coenzyme Q, cytochrome C and oxygen. The enzyme complexes IV and the mobile carriers are collectively involved in the transport of electrons which, ultimately, combine with oxygen to produce water. The largest proportion of the oxygen supplied to the body is utilized by the mitochondria for the operation of electron transport chain. Components and reactions of the electron transport chain. There are 5 distinct carriers that participate in the electron transport chain, etc. Carriers are sequentially arranged, Figure 7, and are responsible for the transfer of electrons from a given substrate to ultimately combine with proton and oxygen to form water. Nicotinamide. Of the 2 coenzymes NAD+ and NADP+ derived from the vitamin niacin, NAD+ is more actively involved in the etc. NAD+ is reduced to NADH plus H+ by dehydrogenases with the removal of 2 hydrogen atoms from the substrate, O2. The substrates include glyceraldehyde 3-phosphate, pyruvate, isocitrate, alpha-ketoglutarate and malate. O2 plus NAD+ if and only if A plus NADH plus H+. NADPH plus H+ produced by NADP plus dependent dehydrogenase is not usually a substrate for etc. NADPH is more effectively utilized for anabolic reactions, e.g. fatty acid synthesis, cholesterol synthesis. 2. Flavoproteins. The enzyme NADH dehydrogenase NADH coenzyme Q reductase is a flavoprotein with FMN as the prosthetic group. The coenzyme FMN accepts 2 electrons and a proton to form FMNH2. NADH dehydrogenase is a complex enzyme closely associated with non-heme iron proteins, NHI, or iron sulfur proteins, FEs. NADH plus H plus FMN7 NAD plus FMNH2. Succinate dehydrogenase, succinate coenzyme Q reductase, is an enzyme found in the inner mitochondrial membrane. It is also a flavoprotein with FAD as the coenzyme. This can accept 2 hydrogen atoms, 2H plus 2E, from succinate. Succinate plus FAD fumarate plus FADH2. 3. Iron proteins. The iron sulfur FEs proteins exist in the oxidized Fe3 plus or reduced Fe2 plus state. About half a dozen FEs proteins connected with respiratory chain have been identified. However, the mechanism of action of iron sulfur proteins in the etc. is not clearly understood. FEs participates in the transfer of electrons from FMN to coenzyme Q. Other FEs proteins associated with cytochrome B and cytochrome C participate in the transport of electrons. 1 FEs participates in the transfer of electrons from FMN to coenzyme Q. Other FEs proteins associated with cytochrome B and cytochrome C participate in the transport of electrons. IV. Coenzyme Q. Coenzyme Q is also known as ubiquinone since it is ubiquitous in living system. It is a quinone derivative with a variable isoprenoid side chain. The mammalian tissues possess a quinone with 10 isoprenoid units which is known as coenzyme Q10 CoQ10. Coenzyme Q is a lipophilic electron carrier. Electrons from FMNH2 produced in the etc. by NADH dehydrogenase or FADH2 produced outside etc. e.g. succinate dehydrogenase, acyl-CoA dehydrogenase. Coenzyme Q is not found in mycobacteria. Vitamin K performs similar function as coenzyme Q in these organisms. Coenzyme Q has no known vitamin precursor in animals. It is directly synthesized in the body. These cytochromes. The cytochromes are conjugated proteins containing heme group. The latter consists of a porphyrin ring with iron atom. The heme group of cytochromes differ from that found in the structure of hemoglobin and myoglobin. The iron of heme cytochromes is alternately oxidized Fe3+ and reduced Fe2+, which is essential for the transport of electrons in the etc. This in contrast to the heme iron of hemoglobin and myoglobin, which remains in the ferrous Fe2+ state. 3 cytochromes were initially discovered from the mammalian mitochondria. They were designated as cytochrome A, B and C depending on the type of heme present in the respective absorption spectrum. Additional cytochromes such as C1, Bl, B2, A3 etc. were discovered later. The electrons are transported from coenzyme Q to cytochromes in the order B, C1c, A and A3. Property of reversible oxidation reduction of heme iron Fe2+ tilde Fe3+ present in cytochromes allows them to function as effective carriers of electrons in He. Cytochrome C, mol, WT. 13,000 is a small protein containing 104 amino acids in a heme group. It is a central member of etc. with an intermediate redox potential. It is rather loosely bound to intermitochondrial membrane and can be easily extracted. Cytochrome N A3. The term cytochrome oxidase is frequently used to collectively represent cytochrome N A3, which is the terminal component of He. Cytochrome oxidase is the only electron carrier, the heme iron of which can directly react with molecular oxygen. Besides with iron, this oxidase also contains copper that undergoes oxidation reduction, Cu2+, Cu+, during the transport of electrons. In the final stage of HC, the transported electrons, the free protons and the molecular oxygen combine to produce water. E5. Oxidative phosphorylation. The transport of electrons through the etc. is linked with the release of free energy. Process of synthesizing ATP from ADP and Pi coupled with the electron transport chain is known as oxidative phosphorylation. The complex V, figure 6, of the inner mitochondrial membrane is the site of oxidative phosphorylation. P-O ratio. The P-O ratio refers to the number of inorganic phosphate molecules utilized for ATP generation for every atom of oxygen consumed. More appropriately, P-O ratio represents the number of molecules of ATP synthesized per pair of electrons carried through etc. The mitochondrial oxidation of NADH with a P-O ratio of 3 can be represented by the following equation. NADH plus H plus 1/2 O2 plus 3 ADP plus 3 Pi NAD plus 3 ATP plus 4 H2O. P-O ratio of 2 is assigned to the oxidation of FADH2. Although yet to be proved beyond doubt, some workers suggest a P-O ratio of 2.5 for NADH plus H plus, and 1.5 for FADH2, based on the proton translocation. Oxidative phosphorylation in etc. There are 3 reactions in the HC that are exergonic to result in the synthesis of 3 ATP molecules, figure 7. The 3 sites of ATP formation in etc. are 1. Oxidation of FMNH2 by coenzyme Q. 2. Oxidation of cytochrome B by cytochrome C1. 3. Cytochrome oxidase reaction. Each one of the above reactions represents a coupling site for ATP production. There are 2 coupling sites for the oxidation of FADH2 P-O ratio 2, since the first site is bypassed. Energetics of oxidative phosphorylation. The transport of electrons from redox pair NAD plus NADH EO equals -0.32 to finally the redox pair 1/2 O2 H2O EO equals plus 0.82 may be simplified and represented in the following equation. 1/2 O2 plus NADH plus H plus H2O plus NAD plus. The redox potential difference between these 2 redox pairs is 1.14 volts, which is equivalent to an energy 52 cal per mole. 3 ATP are synthesized in the etc. when NADH is oxidized, which equals to 21.9 cal, each ATP equals 7.3 cal. The efficiency of energy conservation is calculated as 21.9 x 100 equals 42%. 52. When NADH is oxidized, about 42% of energy is trapped in the form of 3 ATP and the remaining is lost as heat. Mechanism of oxidative phosphorylation. Several important among them, namely, chemical coupling, conformational coupling and chemiosmotic, are discussed below. Chemical coupling hypothesis. This hypothesis was put forth by Edward Slater, 1953. According to chemical coupling hypothesis, during the course of electron transfer in respiratory chain, a series of phosphorylated high-energy intermediates are first produced which are utilized for the synthesis of ATP. These reactions are believed to be analogous to the substrate-level phosphorylation that occurs in glycolysis or citric acid cycle. However, this hypothesis lacks experimental evidence, since all attempts, so far, to isolate any one of the high-energy intermediates have not been successful. Conformational coupling hypothesis. Paul Boyer proposed this hypothesis in 1964. As a consequence of electron transport, a conformational change in the membrane protein is induced. This is believed to be responsible for the synthesis of ATP. The protein attains a high-energy conformation due to electron transfer which, when reverted back to random state, low energy, results in the production of ATP from ADP plus Pi, figure 8. There is some evidence demonstrating conformational changes in mitochondrial membrane proteins. However, there is no conclusive proof that such changes may be actually responsible for ATP synthesis. Chemiosmotic hypothesis. This mechanism, originally proposed by Peter Mitchell, 1961, is now widely accepted. It explains the transport of electrons through the respiratory chain is effectively utilized to produce ATP from ADP plus Pi. The concept of chemiosmotic hypothesis is comparable with energy stored in a battery separated by positive and negative charges. Proton gradient. The inner mitochondrial membrane, as such, is impermeable to protons H+ and hydroxyl ions OH. Transport of electrons through etc. is coupled with the translocation of protons H+ across the inner mitochondrial membrane, coupling membrane, from the matrix to the intermembrane space. The pumping of protons results in an electrochemical or proton gradient. This is due to accumulation of more H+ ions, low pH, on the outer side of the inner mitochondrial membrane than the inner side. The proton gradient developed due to the electron flow in the respiratory chain is sufficient to result in the synthesis of ATP from ADP and pi. Oxidation reduction OR loops or redox loops. It is postulated that the etc. is folded into 3 oxidation reduction loops corresponding to complex I, 3, and IV. Each loop is a coupling site and consists of a hydrogen carrier and an electron carrier. 6 protons are translocated for each electron pair transferred from NADH plus H plus to oxygen. The 3 OR loops, along with the carriers of protons and electrons, are depicted in Fig.11. The first pair of protons 2H plus are translocated via FMN and the second 2H plus via coenzyme Q. There is some controversy regarding the carrier of the third proton pair, designated as X in the Fig.11. Some workers believe that coenzyme Q is involved here. Coenzyme Q is a lipid-soluble small molecule and can freely move across the inner mitochondrial membrane. A cyclic process referred to as coenzyme Q cycle is said to be operative for translocation of protons at 2 OR loops. N system for ATP synthesis. ATP synthase, present in the complex V, utilizes a proton gradient for the synthesis of ATP. This enzyme is also known as ATPase since it can hydrolyze ATP to ADP and pi. ATP synthase is a complex enzyme and consists of 2 functional subunits, namely F1 and F0, figure 10. Its structure is comparable with lollipops. The protons that accumulate on the intermembrane space re-enter the mitochondrial matrix leading to the synthesis of ATP. This process, involving ATP synthase, occurs in 3 stages. 1. Subunit F0 of ATP synthase translocates protons re-entry into the matrix. 2. F1 subunit catalyzes the formation of ATP from ADP and pi. This is believed to involve a conformational change in FL. 3. FO and 1 subunits coordinate with each other and couple the disappearance of proton gradient with ATP synthesis. Evidence in support of chemiosmotic hypothesis. Many experimental observations support the chemisosmotic hypothesis. 1. The intact inner mitochondrial membrane is responsible for oxidative phosphorylation. 2. The inner mitochondrial membrane is impermeable to protons H+, and other ions OH, K+, Cl, etc. 3. ATP can be generated by increasing the H+ ion concentration by adding acid on the outside of mitochondria. 4. Certain enzymes which increase the permeability of inner mitochondrial membrane to protons inhibit ATP synthesis, e.g. 2,4-dinitrophenol. Disorders of oxidative phosphorylation. It is estimated that about 100 polypeptides are required for oxidative phosphorylation. Of these, 15 are coded by mitochondrial DNA, mtDNA, and synthesized in the mitochondria, while the rest are produced in the cytosol coded by nuclear DNA and transported. mtDNA is maternally inherited since mitochondria from a sperm do not enter the fertilized ovum. Mitochondrial DNA is about 10 times more susceptible to mutations than nuclear DNA. mtDNA mutations are more commonly seen in tissues with high rate of oxidative phosphorylation, e.g. central nervous system, skeletal and heart muscle, liver. Leber's hereditary optic neuropathy is an example for mutations in mtDNA. This disorder is characterized by loss of bilateral vision due to neuroretinal degeneration. Inhibitors of electron transport chain. Many site-specific inhibitors of etc. have contributed to the present knowledge of mitochondrial respiration. Selected examples of these inhibitors are given in Figure 7. The inhibitors bind to one of the components of etc. and block the transport of electrons. This is the accumulation of reduced components before the inhibitor blockade step and oxidized components after that step. The synthesis of ATP phosphorylation is dependent on electron transport. Hence, all the site-specific inhibitors of etc. also inhibit ATP formation. 3 possible sites of action for the inhibitors of etc. are identified. 1. NADH and coenzyme Q, fish poison rotenone. Barbiturate drug amytal and antibiotic pyricidin inhibit at this site. 2. Between cytokine and C1, antimycin A and antibiotic, British anti-leukocyte FAL, an antidote used against Wargas are the 2 important inhibitors of the site between cytochrome B and C1. 3. Inhibitors of cytochrome oxidase, carbon monoxide, cyanide, hydrogen sulfide and azide effectively inhibit cytochrome oxidase. Carbon monoxide reacts with reduced form of the cytochrome while cyanide and azide react with oxidized form. Inhibitors of oxidative phosphorylation. Uncouplers. The mitochondrial transport of electrons is tightly coupled with oxidative phosphorylation, ATP synthesis. In other words, oxidation and phosphorylation proceed simultaneously. There are certain compounds that can uncouple or delink the electron transport from oxidative phosphorylation. Such compounds, known as uncouplers, increase the permeability of inner mitochondrial membrane to protons H+. Thus, an uncoupler allows the electron transport but blocks the establishment of proton gradient across the inner mitochondrial membrane. The result is that ATP synthesis does not occur. The energy linked with the transport of electrons is dissipated as heat. Uncouplers allow, often at accelerated rate, oxidation of substrates via NADH or FADH2 without ATP formation. The uncoupler, 2,4-dinitrophenol DNP, has been extensively studied. It is a small lipophilic molecule. DNP is a proton carrier and can easily diffuse through the inner mitochondrial membrane. The other uncouplers include dinitrocresol, pentachlorophenol, trifluoro, carbonyl cyanide phenylhydrazone, FCCP. Thus compound FCCP is said to be 100 times more effective as an uncoupler than dinitrophenol. When administered in high doses, the drug aspirin acts as an uncoupler. Physiological uncouplers. Certain physiological substances which act as uncouplers at higher concentration have been identified. These include the hormone thyroxine and long-chain free fatty acids. The unconjugated bilirubin is also believed to act as an uncoupler. This is, however, yet to be proved beyond doubt. Significance of uncoupling. Uncoupling of respiration from oxidative phosphorylation under natural conditions assumes biological significance. The maintenance of body temperature is particularly important in hairless animals, hibernating animals and the animals adapted to cold. These animals possess a specialized tissue called brown adipose tissue in the upper back and neck portions. The mitochondria of brown adipose tissue are rich in electron carriers and are specialized to carry out an oxidation uncoupled from phosphorylation. This causes liberation of heat when fat is oxidized in the brown adipose tissue. The presence of active brown adipose tissue in certain individuals is believed to protect them from becoming obese. The excess calories consumed by these people are burned and liberated as heat, instead of being stored as fat. Ionophores. The term ionophores is used to collectively represent the lipophilic substances that promote the transport of ions across biological membranes. All the uncouplers described above are, in fact, proton ionophores. The antibiotics valinomycin and nigrosin act as ionophores for K+ ions. Both these compounds are also capable of dissipating proton gradient across the inner mitochondrial membrane and inhibit oxidative phosphorylation. Inhibitors of oxidative phosphorylation. Oligomycin. This antibiotic prevents the mitochondrial oxidation as well as phosphorylation. It binds with the enzyme ATP synthetase and blocks the proton H+ channels. It thus prevents the translocation re-entry of protons into the mitochondrial matrix. Due to this, protons get accumulated at higher concentration in the intermembrane space. Electron transport respiration ultimately stops, since protons cannot be pumped out against steep proton gradients. The action of oligomycin indicates that oxidation and phosphorylation are tightly coupled processes. Atractyloside. This is a plant toxin and inhibits oxidative phosphorylation by an indirect mechanism. The presence adequate concentration of ADP and Pi in the mitochondrial matrix and the transport of ATP into the extramitochondrial system are needed for phosphorylation to proceed continuously. Adenine nucleotide carrier system facilitates the transport of ATP and ADP. Atractyloside inhibits adenine nucleotide carrier and, thus, blocks the adequate supply of ADP, thereby preventing phosphorylation. Transport of reducing equivalence shuttle pathways. The inner mitochondrial membrane is impermeable to NADH. Therefore, the NADH produced in the cytosol cannot directly enter the mitochondria. Two pathways, namely glycerol phosphate shuttle and malate aspartate shuttle, are operative to do this job. They transport the adenosine equivalents from cytosol to mitochondria and not vice versa. The shuttle pathways are not operative to the same extent in all the tissues. Malate aspartate shuttle predominantly occurs in liver and heart, whereas glycerol phosphate shuttle operates in muscle and brain. Glycerol phosphate shuttle. Cytosolic glycerol 3-phosphate dehydrogenase oxidizes NADH to NAD+. The reducing equivalents are transported through glycerol 3-phosphate into the mitochondria. Glycerol 3-phosphate dehydrogenase present in outer surface of inner mitochondrial membrane reduces fat to FADH2. Dihydroxyacin phosphate escapes into the cytosol and the shuttling continues as depicted in Fig. 12. FADH2 gets oxidized via etc. to generate 2-ATP. 2. Malate aspartate shuttle. In the cytosol, oxaloacetate accepts the reducing equivalents NADH and becomes malate. Malate then enters mitochondria where it is oxidized by mitochondrial malate dehydrogenase. In this reaction, NADH and oxaloacetate are regenerated. NADH gets oxidized via electron transport chain and 3-ATP are produced. This is in contrast to glycerol phosphate shuttle where only 2-ATP are produced. In the mitochondria, oxaloacetate participates in transamination reaction with glutamate to produce aspartate and alpha-ketoglutarate. Aspartate enters the cytosol and transaminates with alpha-ketoglutarate to give oxaloacetate and glutamate. The malate aspartate shuttle is shown in Figure 13. E6. Reactions in high-energy compounds, energy-rich compounds. Metabolism is essentially a linked series of chemical reactions that begins with a particular molecule and converts it into some other molecule or molecules in a carefully defined fashion. There are many such defined pathways in the cell, and we will examine a few of them in some detail later. These pathways are interdependent, and their activity is coordinated by exquisitely sensitive means of communication in which allosteric enzymes are predominant. We can metabolic pathways into two broad classes: 1. Those that convert energy into biologically useful forms, and 2. Those that require inputs of energy to proceed. Although division is often imprecise, it is nonetheless a useful distinction in an examination of metabolism. Those reactions that transform fuels into cellular energy are called catabolic reactions or, more generally, catabolism. Those reactions that require energy such as the synthesis of glucose, fats, or DNA are called anabolic reactions or anabolism. Useful forms of energy that are produced in catabolism are employed in anabolism to generate complex structures from simple ones, or energy-rich states from energy-poor ones. Pathways can be either anabolic or catabolic, depending on the energy conditions in the cell. They are referred to as amphibolic pathways. A thermodynamically unfavorable reaction can be driven by a favorable reaction. How are specific pathways constructed from individual reactions? A path must satisfy minimally two criteria. 1. The individual reactions must be specific, and 2. The entire set of reactions that constitute the pathway must be thermodynamically favored. A reaction that is specific will yield only one particular product or set of products from its reactants. The function of enzymes is to provide this specificity. The thermodynamics of metabolism is most readily approached in terms of free energy. A reaction can occur spontaneously only if increment G, the change in free energy, is negative. Recall that increment G for the formation of products C and D from substrates A and B is given by. Thus, the increment G of a reaction depends on the nature of the reactant and products expressed by the increment G degree. Term, the standard free energy change, and on their concentrations expressed by the second term. An important thermodynamic fact is that the overall free energy change for a chemically coupled series of reactions is equal to the sum of the free energy changes of the individual steps. Consider the following reactions. Under standard conditions, it cannot be spontaneously converted into B and C, because Dg is positive. However, the conversion of B into D under standard conditions is thermodynamically feasible. Because free energy changes are additive, the conversion of A into C and D has an increment G degree of -3 cal mole -1 -13 kilojoules mole -1, which means that it can occur spontaneously under standard conditions. Thus, a thermodynamically unfavorable reaction can be driven by a thermodynamically favorable reaction to which it is coupled. In this example, the chemical intermediate B, common to both reactions, couples the reactions. Thus, metabolic pathways are formed by the coupling of enzyme-catalyzed reactions such that the overall free energy of the pathway is negative. ATP is the universal currency of free energy in biological systems. Just as commerce is facilitated by the use of a common currency, the commerce of the cell metabolism is facilitated by the use of a common energy currency, adenosine triphosphate ATP. Part of the free energy from the oxidation of foodstuffs and from light is transformed into this highly accessible molecule, which acts as the free energy donor in most energy-requiring processes such as motion, active transport, or biosynthesis. ATP is a nucleotide consisting of an adenine, a ribose, and a triphosphate unit. The active form of ATP is usually a complex of ATP with Mg2+ or Mn2+. In considering the role of ATP as an energy carrier, we can focus on its triphosphate moiety. ATP is an energy-rich molecule because its triphosphate unit contains two phosphoanhydride bonds. Free energy is liberated when ATP is hydrolyzed to adenosine diphosphate ADP and orthophosphate PI, or when ATP is hydrolyzed to adenosine monophosphate AMP and pyrophosphate PPI. The precise DG degree of these reactions depends on the ionic strength of the medium and on the concentrations of Mg2+ and other metal ions. Under typical concentrations, the actual DG for these hydrolyzes is approximately -12 kilocalories mole -1, -50 kilojoules mole -1. Free energy liberated in the hydrolysis of ATP is harnessed to drive reactions that require an input of free energy, such as muscle contraction. In turn, ATP is formed from ADP and PI when fuel molecules are oxidized in chemotrophs or when light is trapped by phototrophs. This ATP-ADP cycle is the fundamental mode of energy exchange in biological systems. Some biotic reactions are driven by hydrolysis of nucleoside triphosphates that are analogous to ATP, namely guanosine triphosphate GTP, uridine triphosphate UTP, and cytidine triphosphate CTP. The diphosphate forms of these nucleotides are denoted by GDP, UDP, and CDP, and the monophosphate forms by GMP, UMP, and CMP. Enzymes can catalyze the transfer of the terminal phosphoryl group from one nucleotide to another. The phosphorylation of nucleoside monophosphates is catalyzed by a family of nucleoside monophosphate kinases. The phosphorylation of nucleoside diphosphates is catalyzed by nucleoside diphosphate kinase, an enzyme with broad specificity. To note that, although all of the nucleotide triphosphates are energetically equivalent, ATP is nonetheless the primary cellular energy carrier. In addition, two important electron carriers, NAD+ and FAD, are derivatives of ATP. The role of ATP in energy metabolism is paramount. ATP hydrolysis drives metabolism by shifting the equilibrium of coupled reactions. How does coupling to ATP hydrolysis make possible an otherwise unfavorable reaction? Consider a chemical reaction that is thermodynamically unfavorable without an input of free energy, a situation common to many biosynthetic reactions. So the standard free energy of the conversion of compound A into compound B is plus 4.0 kilocalories mole -1 plus 13 kilojoules mole 1. The equilibrium constant K. EQ of this reaction at 25 degrees Celsius is related to DG degree in units of kilocalories per mole by. Thus, net version of A into B cannot occur when the molar ratio of B to A is equal to or greater than 1.15 times 10 to 3. However, A can be converted into B under these conditions if the reaction is coupled to the hydrolysis of ATP. The new overall reaction is its standard energy change of -3.3 kilocalories mole -1, -13.8 kilojoules mole -1 is the sum of the value of DG degree for the conversion of A in plus 4.0 kilocalories mole -1 plus 12.6 kilojoules mole -1 and the value of DG degree for the hydrolysis of ATP -7.3 kilocalories mole -1 -30.5 kilojoules mole -1. At pH 7, the equilibrium constant of this coupled reaction is at equilibrium, the ratio of B to A is given by ATP generating system of cells maintains the ATP-ADP-PI ratio at a high level, typically of the order of 500 m-1. For this ratio, which means that the hydrolysis of ATP enables it to be converted into B until the B-A ratio reaches a value of 1.34 times 10^5. This equilibrium ratio is strikingly different from the value of 1.15 times 10 to 3 for the reaction A. In the absence of ATP hydrolysis. In other words, coupling the hydrolysis of ATP with the conversion of A into B has changed the equilibrium ratio of B to A by a factor of about 10^8. See here the thermodynamic essence of ATP's action as an energy coupling agent. Cells maintain a high level of ATP by using oxidizable substrates or light as sources of free energy. Hydrolysis of an ATP molecule in a coupled reaction then changes the equilibrium ratio of products to reactants by a very large factor, of the order of 10^8. More generally, the hydrolysis of NATP molecules changes the equilibrium ratio of a coupled reaction or sequence of reactions by a factor of 10^8n. For example, the hydrolysis of 3 ATP molecules in a coupled reaction changes the equilibrium ratio by a factor of 10^24. Thus, a thermodynamically unfavorable reaction sequence can be converted into a favorable one by coupling it to the hydrolysis of a sufficient number of ATP molecules in a new reaction. It should also be emphasized that A and B in the preceding coupled reaction may be interpreted very generally, not only as different chemical species. For example, A and B may represent activated and unactivated conformations of a protein. In this case, phosphorylation with ATP may be a means of conversion into an activated conformation. Such a conformation can store free energy, which can then be used to drive a thermodynamically unfavorable reaction. Such changes in conformation, molecular motors such as myosin, kinesin, and dynein convert the chemical energy of ATP into mechanical energy. Indeed, this conversion is the basis of muscle contraction. Alternatively, A and B may refer to the concentrations of an ion or molecule on the outside and inside of a cell, as in the active transport of a nutrient. The active transport of Na+ and K+ across membranes is driven by the phosphorylation of the sodium-potassium pump by ATP and its subsequent dephosphorylation. Structural basis of the high phosphoryl transfer potential of ATP. As illustrated by molecular motors and ion pumps, phosphoryl transfer is a common means of energy coupling. Furthermore, phosphoryl transfer is also widely used in the intracellular transmission of information. What makes ATP a particularly efficient phosphoryl group donor? Let us compare the standard free energy of hydrolysis of ATP with that of a phosphate ester, such as glycerol 3-phosphate. The magnitude of increment G degree. For the hydrolysis of glycerol 3-phosphate is much smaller than that of ATP, which means that ATP has a stronger tendency to transfer its terminal phosphoryl group to water than does glycerol 3-phosphate. In other words, ATP has a higher phosphoryl transfer potential, phosphoryl group transfer potential, than does glycerol 3-phosphate. What is the structural basis of the high phosphoryl transfer potential of ATP? Because increment G degree depends on the difference in energy of the products and reactants, the structures of both ATP and its hydrolysis products, ADP and PI, must be examined to answer this question. 3 factors are important: resonance stabilization, electrostatic repulsion, and stabilization due to hydration. ADP and, particularly, PI, have greater resonance stabilization than does ATP. Orthophosphate has a number of resonance forms of similar energy figure, whereas the G-phosphoryl group of ATP has a smaller number. Or unfavorable because a positively charged oxygen atom is adjacent to a positively charged phosphorus atom, an electrostatically unfavorable juxtaposition. Furthermore, at pH 7, the triphosphate unit of ATP carries about 4 negative charges. These charges repel one another because they are in close proximity. The repulsion between them is reduced when ATP is hydrolyzed. Finally, water can bind more effectively to ADP and PI than it can to the phosphoanhydride part of ATP, stabilizing the ADP and PI by hydration. AT is called a high-energy phosphate compound, and its phosphoanhydride bonds are referred to as high-energy bonds. Indeed, squiggle, tilde-p is often used to indicate such a bond. Nonetheless, there is nothing special about the bonds themselves. They are high-energy bonds in the sense that much free energy is released when they are hydrolyzed, for the aforegiven reasons. ATP, the most important high-energy compound. Adenosine triphosphate, ATP, is a unique and the most important high-energy molecule in the living cells. It consists of an adenine, a ribose and a triphosphate moiety. ATP is a high-energy compound due to the presence of 2 phosphoanhydride bonds in the triphosphate unit. ATP serves as the energy currency of a cell as is evident from the ATP-ADP cycle. ATP-ADP cycle. The hydrolysis of ATP is associated with the release of large amount of energy. AT plus H2O7 ADP plus pi plus 7.3 cal. The energy liberated is utilized for various processes like muscle contraction, active transport, etc. ATP can also act as a donor of high-energy phosphate to low-energy compounds, to make them energy-rich. On the other hand, ADP can accept high-energy phosphate from the compounds possessing higher free energy content to form ATP. AT serves as an immediately available energy currency of the cell which is constantly being utilized and regenerated. This is represented by ATP-ADP cycle, the fundamental basis of energy exchange reactions in living system figure 2. The turnover of ATP is very high. It is estimated that a resting man consumes about 40 kilograms ATP per day. As much as 0.5 kilograms ATP per minute is believed to be spent during strenuous exercise. ATP acts as an energy link between the catabolism, degradation of molecules, and anabolism, synthesis, in the biological system. Synthesis of ATP. ATP can be synthesized in 2 ways. 1. Oxidative phosphorylation. This is the major source of ATP in aerobic organisms. It is linked with the mitochondrial electron transport chain, details described later. 2. Substrate-level phosphorylation. ATP may be directly synthesized during substrate oxidation in the metabolism. The high-energy compounds such as phosphoenolpyruvate and 1,3-bisphosphoglycerate, intermediates of glycolysis, and succinyl-CoA of citric acid cycle can transfer high-energy phosphate to ultimately produce ATP. Certain compounds are encountered in the biological system which, on hydrolysis, yield energy. The term high-energy compounds or energy-rich compounds are usually applied to substances which possess sufficient free energy to liberate at least 7 cal per mole at pH 7.0. Certain other compounds which liberate less than 7.0 cal per mole lower than ATP hydrolysis to ADP plus pi are referred to as low-energy compounds. The list of important high-energy and low-energy metabolites is given in Table 1. All the high-energy compounds when hydrolyzed liberate more energy than that of ATP. The phosphoenolpyruvate, 1,3-bisphosphoglycerate, phosphocreatine, etc. Both high-energy compounds contain phosphate group, exception acetyl-CoA, hence they are called high-energy phosphate compounds. Classification of high-energy compounds. There are at least 5 groups of high-energy compounds. Pyrophosphates e.g. ATP. 2. Acyl phosphates e.g. 1,3-bisphosphoglycerate. 3. Enol phosphates e.g. phosphoenolpyruvate. 4. Thioesters e.g. acetyl-CoA. 5. Phosphogonadines e.g. phosphocreatine. Table 2 gives some more details on the high-energy compounds, including the high-energy bonds present in each category. High bonds. The high-energy compounds possess acid anhydride bonds, mostly phosphoanhydride bonds, which are formed by the condensation of two acidic groups or related compounds. These bonds are referred to as high-energy bonds, since the free energy is liberated when these bonds are hydrolyzed. Lippmann suggested use of the symbol tilde to represent high-energy bond. For instance, ATP is written as AMP_P_P. Storage forms of high-energy phosphates. Phosphocreatine, creatine phosphate, stored in vertebrate muscle and brain, is an energy-rich compound. In invertebrates, phosphoarginine, arginine phosphate replaces phosphocreatine. Biological energy transducers. Photosynthesis. 1. Photosynthesis is essentially the only mechanism of energy input in the living world. Photosynthesis. Light synthesis, putting together, is an anabolic process of manufacture of organic compounds inside the chlorophyll-containing cells from carbon dioxide and water with the help of sunlight as a source of energy. A simple equation of photosynthesis is as follows. Chlorophyll 6CO2 plus 16H2O6H12O6 plus 6O2 radiant energy. However, the function of water is to provide hydrogen for the synthesis of organic compounds, all the liberated oxygen comes from it. Therefore, the equation is modified as chlorophyll 6CO2 plus 12H2O6H12O6 plus 6H2O plus 6O2. Radiant energy. 3. Chloroplast is the seat of photosynthesis and is best exemplified in the higher plants. The external surface of thylakoids contains the photosynthetic pigments and serves the ends of light reaction. The stroma, on the other hand, is concerned with the events of the dark reaction. 4. Photosynthetic. The photosynthetic pigments present in thylakoid membranes consist largely of two kinds of green chlorophylls, chlorophyll 8C55H70O5 N 4 megagrams, and chlorophyll B C55H72O6 N 4 megagrams. Also present are yellow to orange pigments classified as carotenoids. There are two kinds of carotenoids, the pure hydrocarbon carotenes and the oxygen-containing xanthophylls. Carotenoids, especially viola-xanthan, a xanthophyle, also exist in the chloroplast envelope, giving it a yellowish color. In most plants, including green algae, beta-carotene and lutein are the most abundant carotenoids in the thylakoids. 5. Photosynthesis consists of two types of reactions, a light-dependent one and a light-independent one. Light-dependent reaction is a photochemical reaction or light reaction as it came to be called, culminating in the generation of NADPH2, ATP and evolution of molecular oxygen. The ADPH2 and ATP are energy-rich, having caught the electrons that became available when light impinged upon chlorophyll. They form the assimilatory power, utilized for Ca2 fixation. The events of CO2 fixation is light-independent reaction and is designated as dark reaction. 6. Light reactions of two-phase. Phase 1 energy absorption and retention of light by the photosynthetic pigments, and phase 2 energy transduction, conversion of light energy absorbed in phase 1 into chemical energy ATP and NADPH2 by photophosphorylation. 7. Most green plants absorb light in the visible spectrum, 390-700 nanometers, whereas purple bacteria employ wavelengths ranging from near ultraviolet to infrared, 800-950 nanometers. This range of the spectrum through which photosynthesis can take place is called photosynthetically active radiation. But the entire range is not employable in photosynthesis. The green plants, for example, absorb light maximally in the red and blue regions of the spectrum. A study of absorption spectra shows the quantitative relationship between the wavelength of light and its absorption by the pigment in question. Thus, we see that chlorophyll has its absorption peaks at 660 nanometers and 430 nanometers, chlorophyll B at 648 nanometers and 456 nanometers, carotene at 478 nanometers and 449 nanometers, and xanthophyll same as carotene. 8. Photosystems. Photosystem I has been located in the thylakoid membranes. It is made up of three forms of chlorophyll A, one absorbing maximally at 683 nanometers, the second absorbing maximally at 695 nanometers, and the third at 670 nanometers. The last of these has been called P700. Photosystem have been located in the stroma thylakoids. It is made up of two forms of chlorophyll with maximum absorption at 670 and 690 nanometers. The second form is christened P690. Each has three components. A reaction center made up of a special chlorophyll molecule. In photosystem I it is a protein-bound chlorophyll molecule, P700. In photosystem II it is P690. The reaction centers are the actual sites where light energy is converted to chemical energy. Some carriers in photosystem I, X, plastocyanin, cytochrome F and ferredoxin is the electron carrier. Photosystem II has plastoquinone and cytochrome B559. Other chlorophyll and carotenoids, which merely serve to transfer the light absorbed by them to the active centers. Photosystem I takes part in both cyclic and non-cyclic photophosphorylations. PSI can carry on cyclic photosphorylation independently. Normally it drives an electron from photosystem II to NADP+. Photosystem II picks up electron release during photolysis of water. The same is extruded on absorption of light energy. As the extruded electron passes over cytochrome complex, sufficient energy is released to take part in the synthesis of ATP from ADP and inorganic phosphate. This photophosphorylation is non-cyclic. PS2 can operate only in conjunction with PSI. 9. The excited molecules of P700 and P690 transduce their energies to generate ATP and NADPH2. Molecular oxygen is also produced, but it escapes out of the photosynthetic system. ATP and NADPH2 together constitute the assimilatory power and are employed in the fixation of CO2 in the dark reaction. 10. The oxygen that is evolved during photosynthesis comes from water and it is part of the photoact2 mediated by PSII. Another aspect of oxygen evolution during photosynthesis is its relationship to the presence of certain ions in the medium such as Cl, Mn2+ and bicarbonate. 2H2OO2 plus 4H plus 4E. 11. The production of dioxide to a carbohydrate is the essence of this phase, and this is accomplished through the employment of the assimilatory power ATP and NADPH2 generated in the light reaction. This energy stabilization is a dark reaction. The enzymes required for the process are present in the matrix or stroma of the chloroplast. There are two MAM pathways for the biosynthetic or dark phase Calvin cycle and C4 dicarboxylic acid cycle. The plants exhibiting the two are respectively called C3 and C4 plants. Calvin cycle. 12.